Alright, for today's lesson, you will need to download 3 files and load them into your Linux virtual machine. Alright, as you can see here on my screen, I have downloaded the file for this week called My Bioeditor, c3.fasta, and I have a copy of our textbook. Now, you don't have to actually download the copy of the textbook. That should be on your system. You may have a physical copy. I'm just going to use it to illustrate a topic, something called CPG Islands today. It is in your textbook, so I'm going to kind of review it, show you how to search the textbook for some information about what we're talking about. All right, now the other 2 files are a Python program which I have written, and you'll need to download this My Bioeditor. It should be in this week's folder. All right, along with the C3 FASTA file, which we've already investigated C3 files. FASTA files. Now, we've already looked at Python. All right, so you already know how to use the Python editor spider, which stands for Scientific Python Editor. And we've done a little bit of sequence analysis where you've learned about FASTA files. And we're going to begin what we call annotation today. And we're really not annotating. Annotation is kind of like finding the punctuation for a gene or for a sequence. Maybe you've sequenced a piece of DNA and you basically want to find out if there's some genes there, and if they're there, what kind of functionality they might have. Basically, it's like punctuation from some type of text. Just like you have capital letters and periods and spaces and commas, you have similar things within genes, sequences that have some function. We're going to begin this with something called CPG Islands. So let's investigate CPG Islands using our textbook. So I'm going to open our textbook here, and what I've done is I have gone up here to the search, edit, find, all right, and I've typed in CPG, and sure enough I get a whole bunch of references to CPG within the textbook. And the page I'm actually looking for here is page 693, and here it says CPG Islands represent an example of a regulatory element. This is the punctuation I'm talking about. And now I also want to reference that later on we will be using something called the UCSC Genome Browser, which kind of does a lot of what we're going to do today for us. It finds CPG islands, transcription factors, but we need to kind of do it by hand a little bit, just so that you know what the computer is actually doing for you. Normally you use a computer like something like the UCSC Genome Browser, it just gives you an answer. This is going to tell you, kind of show you how you got that answer. CPG islands actually are regulatory elements that you usually find at the beginning of a gene. So it's a good way to locate a gene. C's followed by G's are actually rare within the genome, and you'll need to know why. That's in the textbook. But you'll also need to know what the regulatory function is. That's also in the textbook, and we'll also talk about that a little bit more in another lecture. But for us to do that, we need a sequence, and I have a sequence from chromosome 3 in humans. Now, something like flies don't have CPG islands, but humans do, and you usually find them, like I said, at the beginning of a gene. So we're going to use My Bioeditor, which is a Python program. It's one that I have written, and we're going to use Spider. All right, we're going to open up our program. We're going to hit the little green arrow that we've done before, and we're going to run the program. You may have seen me use this before in class. You now have your own copy of it if you wish to use this. Now, what this program is going to do, it's really no more than a text editor. But it's a text editor I've written that has some functions, all right, that are similar to other editors, but they display it in such a way that we can kind of look at the sequence in a way that I like to look at it, and it's actually visually appealing on something like this video. So let's open our sequence, and here we have it, and we actually get to see the whole sequence all at once. It's about 8,000 nucleotides, and what we're going to do is something very simple. We're going to look for a very rare sequence, a C followed by a G. Wherever you find these C's followed by G's, you typically know that there's something interesting going on. Probably there is a gene nearby. So let's do a search. I'm going to go down here to search. All right, and basically we have three different colors we can choose from. I'm going to choose, oh, since it's flashing here, blue, and I am going to type in C followed by G. And I am going to hit the highlight blue subsequence here, and here we get some statistics, all right, about where they are. It's just a list of where you find these C's followed by G's. All right, and we also have a nice little graphic here showing you where they are. Now, the interesting thing here is we see that up here, we don't get very many C's followed by G's. Down here, we get a ton of them, all right, and especially in this region. And this is what we would call a CPG island. Now, the interesting thing and what I like to do with this program is I like to change the shape, all right, of my window. And I believe this gives me personally a better view of where the interesting things are. So I can make it wide, I can make it narrow, and you can see that there's not many C's followed by G's in the early part of this sequence. But down here in the middle, it sure does seem to have a lot of it. So we might take note that this region here, whatever it is, all right, is of interest. Maybe between here and here, as you can see, I've highlighted it. And we could copy that part of the sequence. So I can go like Control C here, and maybe I can put this into a different text editor. Now, to tell you the truth, you could use any text editor, all right, to do what I just did, except for I get to do multiple sequences and color and things like that. So here's our sequence. I could make a FASTA out of it. Many things I could do to analyze our sequence. But this is our first step in actually looking at annotating a gene, and we get introduced here to a little editor that I think is very useful for the type of thing that we're going to be doing. All right, and it also is something that those of you who are programmers might be interested in. We're going to do a project, our third project, which is more individualized. And if you want to do a programming project, I would like to add some features to this program. It's graphical and it's kind of cool. It's nice and visual, but it needs some more analytical tools, and I'd like to get somebody to write some code for me and improve some things. I'd like to add dot plots, which we've looked at before, to this program. If somebody's interested in doing that, please contact me, and we'll see about somebody using this as their project. All right, well, that's it for today. This kind of introduces you to basically this little program along with CPGs. There's an assignment this week for you to download some sequences and look for CPG islands. It's not a hard assignment. Basically, we're just beginning our annotation part of the class, and you need to learn to search and find things within these sequences. And that's it.